Audio Jungle.
The big question I want to ask is, I feel, the question of the day, which is about immunotherapy. And it's been hailed as a major breakthrough, as have many other major breakthroughs. But we're only actually seeing response rates of 20 to 30%. And to everyone except Jay, I wanted you to just answer, what is it going to take to address the non-responders? Elad, do you want to have a go at that?
Sure. Yeah, so I think that, for one, we're meeting today actually around the same time as the European Society of Medical Oncology, where they are also reporting some results of recent clinical trials, including an NCI-sponsored clinical trial of Merkel cell cancer. It was a rare cancer where there was a 71% response rate in a small number of patients. So I think that there is some variability even with PD-1 inhibitors across different histologies that we're seeing. We're seeing some differences also with regard to combination therapy, where we, of course, have seen with ipilimumab and nivolumab sometimes higher response rates with the immune checkpoint inhibitors that are approved in melanoma from BMS. I think that what we've lacked to a large degree in immunotherapy to date are good predictive animal models. So we have been in something of a curious situation where the information, the data gathering that we're having from the clinical trials that have come out we're actually essentially using the human as the animal model. We're learning directly from patients going back to the bench. So there's a bench to bedside translational component to a lot of the work. And that in and of itself has actually been useful. And I think that to a large degree, that's what we will need to do in order to further advance immunotherapy.
Elad, do you want to, if you Sure. don't look at me, just look at the audience, okay. You may find the microphone picks up a bit better. So just ignore me.
So the
Has it gone off?
Okay.
We've got a little mic problem. All right, we have someone coming to save the day. Ta-da! It's the cavalry. It's okay, don't worry.
Okay. All right, sure. Okay. Yeah. So essentially, I think we're learning a lot from patients in the clinic, and we can have biomarker discovery based on patient samples, and also work to improving those animal models so that we can better predict what we need to do next and better understand the natural history of a tumor. Because to some degree with the immune checkpoint inhibitors, I think there have been a lot of believers in immunotherapy for a very long time. Immunotherapy as a concept is a 100-year-old idea or a mechanism of treating cancers. It just has been a long time until we've gotten to the point of significant success by way of clinically available and widely available therapies. So we obviously were learning a lot from the existing patients, and we need to learn more from the tumor microenvironment, from repeated tumor biopsies, and also understanding how the immune system on its own interacts with cancer.
Jalal, do you want to go ahead?
Absolutely. And I think, first of all, great to be here. But I would like us as a community, I think, to really stop for a second and celebrate and recognize that we are in absolutely a paradigm shift in the treatment of cancer. This is absolutely unprecedented time. And that shift happened in 2013. And 2013 is for the first time where we were absolutely able to show, or in the research area, that basically having the immune system to attack cancer was not just going to be, which was the dogma, it's going to be only in niche indication like melanoma and renal cancer, is when those responses started happening in nasty cancers like lung cancer. I think that opened up completely the whole field of saying every tumor, it is possible, because now I think we've heard the whole morning how much how heterogeneous these cancers are, how every cancer is multiple cancers, and it's for the first time we're actually seeing progress. We're not attacking just the tumor, but actually dealing with the host, and that's for the immune system. So I think that's absolutely a paradigm shift that's happening. I am absolutely glad it's happening in my lifetime. I never thought it would happen in my lifetime. So with all that, though, I think we are seeing, what's exciting is we are seeing responses in multiple tumor types, like Merkel cell, exactly. There was one exciting this weekend in ESMO. But when we look at then the numbers, we see that we are going 20 to 30% or 15 to 23% in lung. Now with the combinations trying to increase that. But for me, that's really just that we understand that this is just the beginning. We're just scratching the surface. We need to understand a lot more, why other patients are not responding. We're already understanding that we are touching just the T cells. We're touching just one element of the immune system. We need to understand all of that. And I actually looking at every patient, what's wrong, how to kickstart that, what the immune system does really well, which is attack cancer.
Edward.
Yeah, let me also add my thanks for the opportunity to participate in this panel. I agree with what I've heard already from the other panelists. Would just add some other observations, acknowledging that my field of interest and expertise is red blood cells, so I tread in the immunological ice very, very carefully. But I think we need to understand, I actually would turn the question around. I was kind of amazed that 20 to 30% of patients are responding to these agents, given that basically there's two pathways we've attacked. With CTLA-4 and the PD-1, PD-L1 circuits, they're just two of literally hundreds of circuits that determine the ultimate immune response and the inflammatory response that follows on it to the benefit or the detriment of the patient. So it is a paradigm shift, but we're just at the very beginning of it. This is, I would roughly analogize this to where we were when we had penicillin and streptomycin in the antibiotic world. And that what we really need to do is to continue to understand the basic immunobiology and immunopathology of cancer. There are already many, many other targets and potential pathways that people are exploring. So I think we need to broaden the number of agents that can modify the T cell response, but in addition recognize that NK cells, the antibody response, all need to be done. I think another thing that's clear from the development of the CAR T cells, and remember the success with CAR T cell therapy and engineered T cells really has been most successful, again, focusing on just a couple of neoantigens. That we have got these two arms that I think could converge and increase the response rate. On the one hand, we're trying to activate the latent desire of the, if you will, of the immune system to attack the cancer. On the other hand, we're trying to provoke a new immune response by creating an immune response to neoantigens. Combining those two could be very powerful. I think the limited efficacy of cancer vaccines is in part due to the same mechanisms that we're dealing with. So that, to me, combination chemotherapy in the future is a combination of immune strategies plus the addition of targets against driver mutations. That we're going to have to get to that part. And I'm sure we'll be talking about the challenges of actually doing that in a clinical setting with identifying the right patients.
Well, I mean, that is the next question, isn't it? As also is, with all these potential options, is it possible that can we get better at predicting which ones are going to work? Because we have many therapies at our disposal. They need to be stacked, they need to be combined. Do we really have to wait to do one-on-one trials in the old way that we've always done that? Or is there some smarter way that we can be assessing which combinations work? Do we need more science to understand that first, or can we use some kind of cell technology to predict, how to stack immunotherapies? Any ideas?
I think that there are a lot of options out there. One area that's been somewhat encouraging actually has been looking at the number, the number of mutations that given tumors express. So not necessarily the number of mutations that they have a priori, but which ones actually get translated into mutated proteins that ultimately can potentially get recognized by the immune system. And those, that actually is somewhat interesting because there was a Nature paper not too long ago that looked at a variety of different histologies and the numbers of mutations that were seen within those different histologies. And on that list, through sequencing, we were able to determine that melanoma and lung cancer, amongst major histologies, actually had, on average, very high rates of mutation. Renal cell cancer, which actually seems to respond, and also was reported at ESMO this week with nivolumab, one of the PD-1 inhibitors from BMS, that didn't have such a high mutation rate, but what it did have, or seems to have, is a high level of translation from those mutated genes to proteins. And that's potentially indicative of one of the mechanisms by which the immune system recognizes that there's something aberrant that needs to be dealt with. So that's certainly one way. There's also an interest, I know Genentech has been focused on different markers, whether or not they're on other PD-L1 on the immune cells as opposed to on the tumor cells. There are questions as to whether PD-L2 is potentially, so another ligand for the PD-1 inhibitors may actually have some relevance. So there's a lot being explored, and obviously we need to do more.
Does anyone else want to tackle that question?
Yeah, I can address that. I completely agree, and I think the mutational load is definitely one area where people are looking at how you do that. I think we have to also recognize that the advancement in the whole genomics that happened, and knowing exactly, which gene is mutated in this tumor or not, and things like that, are really allowing us to go, and combine these agents and address the question. I think, unfortunately, every time you think we're just going to go and just put them together, we still have to go through, I think what we heard from the previous panel, have to do the clinical trials to address the hypothesis. I think the hypotheses are absolutely there, and it's the genomics becomes almost a no-brainer that we do. But how is that going to be, which patients and how it is going to be in immunotherapy is a whole field that we have to start now, really.
Let's move to precision medicine, because I'm keen to draw Jay into the discussion. Jay, and I wanted to ask you what some of the challenges might be in bringing sequencing technology to the sort of precision applications in cancer. We're talking about immuno-oncology is a particularly difficult example, but there are many other examples.
Sure. Let me begin by thanking you as well for having me here today. There's a whole host of challenges that we're facing in bringing this to the market broadly. When we think about this market, it's not just the large cancer institutes, because we all know that the vast majority of cancer patients are treated in community hospitals where the challenges are vastly different. And we think about this as a global problem, not just a US problem. So in that context, we're trying to bring products, obviously, for the research field here to improve our understanding of the mutational load in cancer, and that's relatively straightforward as we've continued to improve the overall performance and cost points of doing large-scale sequencing. But in the diagnostic sector, the challenges are much greater. Just randomly today, we announced our first product we call the TrueSight Tumor 15. It's got 15 actionable genes on it. We'd love that to be a diagnostic product, but in the US, we can't do that. So we've put this into the market. We can only market it as a research use only product, but it has all the genes on it that are associated with today's marketed therapies. So it would allow, if we could market it as a diagnostic, somebody to run a single panel very inexpensively and get all of the mutations for any marketed therapeutic. Because of the ambiguity in the LDT versus the IVD rules, we don't know exactly whether we're going to put that through the FDA as an IVD or not, because by the time we get done with that process, what we know is actionable genes are going to be dramatically different. And so there's a cost-benefit challenge here as to whether we put in the effort and the cost to get this as an approved product so we could market it broadly to community hospitals and outside the US. In other countries of the world, it's much easier to do this. So we'll probably take this through CE-IVD approval in Europe. It'll be much easier to market this in places like China and India than it will be to do in the US. Having said all that, we recognize the challenges that the FDA has in making sure that these products are safe and they're efficacious. And so we're trying to deal with that challenge, number one. Reimbursement is clearly another whole discussion about how we get these products reimbursed. And we think setting standards around how the sequencing gets done is one way to try to help the payers figure out how to reimburse these products, because there are so many and such a proliferation of different types of sequencing panels at different depths with different genes that the payer problem is a real challenging one.
I mean, how do you see that market evolving? I mean, I think this is an amazing point. It's like there are all these diagnostic tests, and obviously you're Illumina, so you have a certain position. But there's hundreds, if not thousands, of companies that are trying to do this. I mean, how are we going to sort this out? Are we just going to let the free market decide, or are we going to impose some form of order on it? Are we just going to?
Well, I think some form of order is appropriate at some point. I think right now we're still in the very early phases of this, and we're learning about what the right content ought to be, how deeply you need to sequence, what the reports ought to look like, what's actionable and what's not. We're learning so much more about the mutation spectrum of these heterogeneous tumors that I think it's a little early for us to try to put some order into the marketplace. We need to kind of cast a wide net here, let lots of products evolve in the marketplace, understand which ones are the best, but we all know over time this does need to become much more standardized so that we're able to share data much more easily, and report in one physician's office looks much more like what they would get if they were in a different cancer center. That might take us 5+ years to do that, but I think that's the direction we need to head.
Why are not more diagnostic tests approved at the same time as the drug that they're used for? Why does that not happen more often? Does anyone want to tackle that question?
I think the problem is that it happens too often, I think, at the same time. Because if you look at in the area of immunotherapy right now, there is one diagnostic test or predictive marker or whatever you want to call it, that's the actual PD-L1 staining. And we have 4 tests. It's really hard to compare because you don't know the sensitivity of one or the other and everything. And we're all have to evolve into a different model. I don't know which one, but I think, go ahead.
I think what I'm asking you is, when you develop a drug, why don't you at the same time approve the diagnostic that goes with it?
Oh, so we actually, most of the time when we are doing a drug development, we are bringing the diagnostic along. So we work with diagnostic companies to bring that. It's not an easy thing to do. First you have to discover that diagnostic or that biomarker, and then you have to do it in a way that is happening at your phase 1 and phase 3. It's a little bit more challenging in cancer because you're actually going sometimes from phase 1 to phase 3 directly, so you don't even have that time, which is a good thing. It's not a bad thing. So it's not, sometimes the science is not there to get that at the same time.
Right. Okay, but genomics is something that's sort of much more embedded now in your clinical trials, would you say?
Absolutely, absolutely.
Right. Do you want to just describe a little bit about how that's different from the past?
Yeah, I think the whole precision medicine and the fact that we can sequence, I think that was shown today, that you can, thanks to the Illumina of the world and others, that you can, the genomic sequencing or the sequencing is not an issue anymore. It doesn't take, I don't know how many months or whatever. It takes, it's very short and it doesn't cost as much. You're definitely getting those biopsies and you want to learn as much as you can from the tumors because you want to go very, very precise. But like I said, that's now in the whole immunotherapy is we have to start over again. Where are we going to look? Yes, there is the tumor, but what are other things we're going to look at? There is still one single area that's of interest is the whole liquid tumors, because for everything we're talking about, you still have to go and get the biopsy from the patient, which is not always easy. And so I think there is a whole other area that Illumina and others are getting into for liquid tumors, basically a blood test that you can take.
Liquid biopsies, right, as
Yes.
well. I've heard that called
liquid
as well.
biopsies.
And I mean, that presumably is going to allow us to sort of trace the evolution of a cancer as well, rather than just, taking a biopsy once, and we know that it's going to evolve in your body. So that too could kind of transform the way that we view cancers, right?
Yeah, no, absolutely. I think it's a very, very exciting area. I think it's still at its infancy, I would say, but we've made a huge progress there. I think it's going to be fantastic if you really can have, the tumor sheds DNA, and if you can get that DNA and tells you either detect the cancer very early, because we all know that detection very early is very important, but also if it's responding to treatment or not. We're not there yet, but I think there are encouraging
But Jay, I mean, is that
areas.
something, I mean, presumably if people are having liquid biopsies taken kind of on a regular basis, that's an area that your company would be interested in working in?
It is, and we are working in this area. It's we think, one of the most exciting new opportunities ahead of us in this field. Initially, we think liquid biopsies will be used for doing the molecular characterization of the tumor, and particularly initially in situations where it's very difficult to get the solid biopsy. It will also be used for monitoring treatment, so because you can do this every few days or once a week, you can watch the effect of the treatment if the circulating tumor DNA falls to zero, which is clearly what we'd all hope in a treatment regimen. And you can also use that as a monitoring technique to detect recurrence early. And we had a conversation in some of the prior panels, I think, about combination therapy. These combination therapies may be ones that we want to administer sequentially rather than simultaneously, because we can detect this recurrence with a liquid biopsy way earlier than we'd ever see it, either through symptoms or imaging. And then the holy grail here, of course, is whether we can actually screen healthy patients for cancer. That requires advancements in the technology. It will require extraordinarily deep sequencing, so it will require, we think, reductions in cost and new formats of the arrays, of the testing array that we would produce. But I think this whole area is extraordinarily fascinating, and one that we're putting a lot of effort into. In fact, we just recently announced a collaboration with Memorial Sloan Kettering to get a stream of thousands of patient samples into our lab, so we can do the liquid biopsy and compare that against what they find using their standard sequencing test off the tumor biopsy. We know very little, I would say, at this point about the actual connection of the biology of the tumor with what we see in the blood, and so there needs to be a tremendous amount of clinical work here to make that connection. But interestingly, we're already finding a much greater variation spectrum in the blood than we typically find in the solid tumor, which is incredibly exciting because it gives us the potential, we think, to understand tumor heterogeneity at a much better level than we could ever through the traditional biopsy methods.
Edward, I know you were indicating you wanted to come in.
Yeah, I just thought I'd offer some thoughts from our experience. We've been doing enterprise-level sequence analysis on any of our patients who will consent to have it done. We have to do it right now as a part of a clinical research protocol because there is no reimbursement for it. And in Massachusetts, we have this funny rule where we're not allowed to give anything away. So it's treated as a neuromint. So we set this up as a clinical research test, a project we call Profile. We've had experience now with somewhere between 10 and 20,000 patients who've been fully analyzed and followed for some period of time. And I think there's a few lessons from that that we've learned that are consistent with what my fellow panelists have said. The first is we should not underestimate the logistical challenges in doing this, particularly when we talk about getting outside of the major cancer centers. Like many other of the major cancer centers, we've set up satellite facilities, we've set up partnerships with community oncologists and so forth. There are literally, just for ours, several dozen points of entry. General surgeons, cancer surgeons, gynecologists, that first bit of tissue that you get that's going to be the thing that's analyzed. This is very much a garbage in, garbage out proposition. How that tissue is handled, how the initial morphologic and histologic diagnosis is made, that you're going to, because you have something to, you have to correlate those, the sequencing actually becomes, and I'll say that with great respect, the easy part. And frankly, in many ways, if you look at the total all-in cost, not the most expensive part, and that's going to be increasingly true. So we're going to have to think about standardizing how tissue is obtained, how the pathologists read it, perhaps centralizing pathology reads as we do in clinical trials right now, so that we're all talking about the same kind of tumor when we say this mutation correlates with that and this treatment has this impact on it. Adding increasingly sequence analysis that helps you predict immunologic function and immunologic potential response to therapy. The other thing we've learned, of course, is that DNA sequencing is wonderful. I got tenure on DNA sequencing, so I'll never knock DNA sequencing. But it is the phone book. And when you take multiple core biopsies, which we're doing now in increasing numbers of patients, as are many of the other cancer centers, so that you get 6 or 7 cores, plus you draw blood, you get circulating tumor cells, you get circulating tumor DNA. So you can look at all these things. It's pretty clear that mutational analysis doesn't always predict what the tumor is actually expressing. It's good. It's better than anything we have, and it's the one thing you can deploy on an enterprise-wide level. But that we're going to have to be able to get to the either the proteomic analysis or the RNA-seq analysis that we're able to do on a much wider scale than it's feasible to do now. Because in the few hundreds of patients that we've done and have been done in other centers, you begin to see that the correlation breaks down. And we need to know that because I think that helps us understand when we have a prediction of a response or a non-response and the exact opposite happens with the patient. As Jay said, it helps us understand more of the tumor heterogeneity. And the third lesson I would say we've learned is that an obvious one. The tumor that's killing our patients isn't the one that we diagnose at the time they come in. It's the one that persists after the initial therapy. And that's particularly true with these very potent newer therapies of immunotherapy and targeted therapy. It has a tremendous selective pressure, more so I think than conventional chemotherapy, on the tumor, which greatly simplifies the population for a while, but then it comes back with this enormous heterogeneity. And to Jay's point, I think he was the one who made it, we really need that second way of sampling the tumor shortly after an effective treatment, so we can figure out what we should do when that population is genetically simplified before it blossoms out with this enormous amount of heterogeneity, and then we're kind of stuck.